So the next session is the doctoral thesis defense. The speaker will have 30 minutes for presentation and another 10 minutes for Q&A session. The speaker is Kiran Jalil. Ms. Kiran Jalil. The title is The Structure Study of Photosynthesis Spore and Germinating Using Quick-Freeze Deep Edge Electron Microscopy. So please.
Good morning, everyone. Thank you for having me here. So this is my, today I'm going to present the doctoral thesis and the topic of my thesis is structural study of Bacillus subtilis spore and germination using quick freeze deep edge electron microscope. So first I would like to introduce about the spore forming bacteria. So these are the main spore forming bacteria. They are Clostridia and the Bacillus. Spore forming bacteria belong to the family of firmicutes, and firmicutes are further classified into two classes, the Bacillus 1 and the Clostridium. So the main target of my research is Bacillus subtilis strain 168. So before going to my results, I would like to explain the details of sporulation and germination. Bacillus subtilis sporulation. So what is sporulation? So when a Bacillus vegetative cell come across some kind of harsh environment like high temperature or nutrient deficiency or some kind of radiation, so it underwent some kind of changes and it goes under asymmetrical cell division. So what is asymmetrical cell division? So this is the unequal division of the cell. One cell is long and the short and other cell is short. So the long cell is known as mother cell and the short cell is baby cell. Next is the process is phagocytosis. When the mother cell engulfs the baby cell, and this process is called phagocytosis. As soon as the phagocytosis is completed, so the next step is the formation of forespore. In the formation of the forespore, the two membranes cover membranes surrounds the spore, and this structure is known as forespore. So as soon as the forespore formation is completed, so next step is the beginning of mother cell lysis. So as soon as the mother cell completely lyses, the forespore release into the environment. And this forespore is now a mature endospore because of its complete structure. And when this mature endospore roams in the environment, as soon as it comes across some favorable condition, for example, some nutrients, peptides, or any other thing which can activate the germination process. So what is germination? So germination is the conversion of this endospore back to the vegetative cell. After that, it can continue the cell division or by binary fission, or it can go back to the spore formation, depending on the environment it faces. So the main interest of my research is these two stages, the mature endospore and the germination. So first of all, I would like to introduce about germination also, because this is very important step and it is focus of my study. So this is the main structure of the spore. Spore has many layers. It's a very highly resistant structure with a lot of layers. So main important layer, which is pore, because it's very important because it contains all the DNA, protein, enzymes, and it is highly acidic and it has lower water concentration. And these are the layers which are surrounding it. For example, the coat layer, this one, and this is the cell membrane which is surrounding the core, the core membrane which is surrounding the core, and this is the cortex peptidoglycan of the spore. So what happens is that when it comes across some nutrients, for example some peptides, amino acids, some favorable condition, so it activates the core membrane GR receptors, and then a change occur inside the spore. For example, a lot of cation release, and this calcium DPA, the dipiclonic acid release, and then we can see the partial dehydration of the core. Next step, the process, as the process continues, it goes to the stage 2, where because of the core become dehydrated and these cation release, following the release of the enzyme into the cortex. Those enzymes are very specific to the cortex dehydrolysis, and they degrade the cortex. And there is a loss of resistance and loss of dormancy. Next step is the degradation of the this brown. So next step is the outgrowth, where this core is actually became the new vegetative cell, and it comes out of the outer shell of the spore, and it gave rise to new vegetative cell. So the last step of the germination is the release of the new cell and the coat degradation, complete degradation of the coat layer. So this is the whole mechanism of germination. So for the purpose of visualization of sporulation and germination, I used quick freeze deep edge electron microscopy. So what is quick freeze deep edge electron microscopy? This is a very unique technique, where first what I did is to make my condition, optimize my condition, and then I visualize under the optical microscopy. Once the conditions are confirmed, then I quickly freeze the sample by using special equipment with a cooled copper block like this. And then next step is to transfer the sample to a high vacuumed, high pressure equipment, and where the cell is fractured with a very cool knife. Next step is deep etching, where we etch the sample at low temperature, very low temperature, like minus. And then the next step is we make replica. These replica are formed by the metallic shadowing, for example platinum. And these replicas are then observed under electron microscope. So this is the whole method of quick freeze deep edge electron microscopy. So next I move into my result. So first I did is to observe the endospore structure, and this is how the spore looks in the optical microscopy. First I confirm my condition, and then next I quickly freeze the spores by quick freeze deep edge electron microscopy, and this is the same. These are the spores under quick freeze. When I magnified the image of the spore, I could see an outer layer which is full of hairy, long filamentous structures. So these filamentous structures have very unique and clear dimension, which ranges from diameter 4 to 10 nanometer. So they are very highly filamentous in branch, and I gave them name like hair-like structure. Previously, some researchers also do experiments, and they find out that the spore consists of outer layer known as crust. But this time we were I was succeed to visualize the native structure of this crust. So the structure of this crust is hair-like long filamentous structure. So I gave it a name hair-like structure. Yes. Okay, so next I treat the spore with lysozyme. So lysozyme is a very famous enzyme. It's antibiotic, and also it is used to degrade the cell wall of the bacteria, and it's commonly used in experiments, and especially in gram-positive bacteria, it degrades the peptidoglycan. And this structure, this is the structure of lysozyme. So I mix the lysozyme with the spore for 1 hour and for 1 mg per ml. And this is the result I got. So first, when I observed under optical microscopy, it was really like normal. It looks really normal. But when I observed under the quick freeze, so it shows surprisingly, it shows a very different outer structure like this. This is the field image of the lysozyme treated spores. So when I magnified the spore structure, it looked like this, with the long spikes, these spikes with various length and density. For example, they look like this. They have different length and they have diameter, same diameter but different length. And these are called spikes. And about 75% of the spores show the spikes, and they have length ranges from 160 to 510 nanometer with a density of 40 to 60 per micrometer scale. So previously, some researchers tried the same thing on Bacillus amylophysician, and they find out the same structure. And this time we could see the same structure in Bacillus subtilis under quick freeze deep edge electron microscopy. And I assume that spores are based on these image analysis and image observation. I assume that these spores are resistant to lysozyme. Next, I was interested to visualize the internal structure of the spore. So as quick freeze has a very unique style, it can fracture the spore and we can see the internal structure three-dimensionally. So I tried the fracture process. So this time the process was same till the fracturing, but I skipped the process of deep etching and I moved directly to the replica formation, and we visualized those replica under electron microscope. So this is the field image of the fractured spores. Now when we move to the magnified image, we could see clearly many layers of the spore. For example, when we magnified this image, so we could see the second rodlet layer after the pressed layer, and this is indicated with the brown steric. Then we could see the cortex, which is a peptidoglycan, spore peptidoglycan, which is underneath the rodlet layer. And then last but not least, we could see the main component of the spore, which is the core. Next, I could also see the section images of the spore, and I can clearly see the three-dimensional structure of some membranous structure and also some vesicles. So here is the section image of a spore. I could see this brown steric indicate the rodlet layer, and then we move to the next image. So this shows the other layers. For example, we could see the rodlet layer, which indicated with brown steric and with triangle. These are I think these are the remnants of the rodlet layer. And then outer vesicles, we could see outer vesicles refer to the vesicles outside the core membrane. These are vesicle-like structure outside the core membrane, which indicated with the green triangle. And then we could see the core vesicles indicated with the black triangle. These are inside the core, and then we could see the core membrane, which is indicated with yellow triangle. So these are some more layers which we could see with the help of deep edge electron microscopy. Sorry, not deep edge, quick freeze fractured electron microscopy. So next target, until now I visualized all the layer of the spore, intact spore. So next my target was to isolate the peptidoglycan of the spore. So first we have to know what is the difference between the vegetative peptidoglycan and the spore peptidoglycan. So there is a big difference between the vegetative peptidoglycan composition. For example, they have alternative group, basically the peptidoglycan is composed of glucose plus amino acid. The main glucose are the acetyl muramic acid and the glucose amine, and they are linked together alternatively through a peptide, some peptides, and whereas in the spore peptidoglycan composition, we could see that this group, N-acetyl muramic acid, is replaced with another group called muramic delta lactam. So spore peptidoglycan is clearly different from the composition is different from vegetative peptidoglycan, and because 50% of the N-acetyl muramic acid is replaced with muramic delta lactam. So next, my aim was to isolate peptidoglycan. So first we isolate the vegetative peptidoglycan, done by Vijay Tahara. He isolated this structure of vegetative peptidoglycan. First, this is the optical microscope result of after treated with STS at 95 degrees centigrade for 12 hours. This is the negative staining result of peptidoglycan, which show clearly a sac-like hollow structure, and these are the structure of the vegetative peptidoglycan. Next, our aim was to isolate peptidoglycan of the spore. So for that, I freeze the control sample, means a washed untreated spore. For that purpose, I just washed the spore with water many times and just confirmed the condition of the spore. Then I observed under the negative staining, and then I used quick freeze for the visualization. Next, I treated the spore with STS at 95 degrees centigrade for 12 hours. This is the result by optical microscopy, and we can see that there is some difference in the spore. But the clear difference we can see is there is some the structure of the spore is clearly compact, and there is no kind of additional layer here. We can also see the same effect on the spores under quick freeze deep edge electron microscopy. Next, to confirm that this is peptidoglycan or not, we additionally treated the sample with lysozyme. And we can clearly see the difference in the structure from the previous reaction. They show very hollow structure, sac-like structure. And it was confirmed by negative staining electron microscopy further. And then we observed under deep edge electron microscopy. So first we will look at the magnified image of the vegetative cell plus STS. We treated the vegetative cell with STS, and we see a mesh-like structure here. Then I observed the magnified image of the spore peptidoglycan when the spore were treated with the STS, and it looked like rough structure. It was very different from the vegetative cell. Next, when it was additionally treated with lysozyme, I could see a very clear difference, and it showed a very clear porous structure, and which is the effect of the lysozyme. So conclusion, I assume the conclusion that what happened is that when the spore PG was treated with the lysozyme, it removed some layer, extra layer from the PG, and we could see a porous structure. So for that we should understand how vegetative lysozyme act on vegetative PG, peptidoglycan. So actually it cleaves the beta 1 to 4 glycosidic linkage. It is this linkage between N-acetyl muramic and N-acetyl glucose amine, and it cleaves that and form peptides. But in spore PG, why the spore PG was resistant, I assume that because it is replaced with the NAM is replaced with MAL, muramic delta lactam, so lysozyme can't cleave the bond between these two, and therefore it shows a resistance to lysozyme. Therefore the spore PG shows resistance to the lysozyme. So that's all until the endospore. Now I will focus on the germination of the spore. So I just focus on different stages of the spore, from the spores, from the degradation to the hatching. So I will move step by step. So first I focus on the stage 1, where the spore starts germinating. Then I add the germination buffer inside the spore, and then I check the conditions by using optical microscopy. So initially till 45 minutes, it looks the cells doesn't change under optical microscopy. So I decide to visualize what happened to the actually happening to the germinating spores. So this is the quick freeze field image, and we could see still there are some cells which have the outer hair-like structure. Next, I observe the fractured image of the spore. So this is the fractured image of the germinating spore at 45 minutes. Note that I already have added the germinating buffer inside. So I could see some fractured cells with these type of pores-like structure. This means that the coat layer is degrading. That's result of the germination. So I could see at 45 minutes that crust-like hair, I could see the crust hair-like structure, and also I could see the degradation of the rodlet layer. Next, I visualize the section images of the spore at 45 minutes. So this is the field image of section spores, germinating spores, and this is the magnified image of the germinating spore. Further, when we see the magnified image of the spore, we could see some clear structures. For example, we could see the degrading rodlet layer, which is indicated with a brown triangle. Then we could see the expansion of the core membrane. It's clearly seen in the germinating spore that this yellow triangle indicate the core membrane, and we could clearly see the expansion increase in the area during the germination. Then we could also see some degrading or remnants of the core vesicles. So it means that during the 45 minutes, internally the core expands and these core vesicles are disintegrated. Next, I focus to the next stage, which is the cortex hydrolysis and loss of resistance. But because I am doing the structure analysis, so I focused on the cortex hydrolysis. So at 45 minutes, at 4.5 hour, I freeze the samples at 4.5 hours. So first I confirm the condition of the sample. So because the germination is a process in which the spores don't germinate at the same time. So this condition was the best condition where I can see all the stages at the same time. So I choose this condition, and I could see many interesting images. This is the field image of the spore, germinating spore at 4.5 hours. So this is the magnified image of one of the germinating spore. I could see very clearly this is the rodlet outer second layer, and then I could see the degradation of the cortex during the germination. So this is the magnified image of the cortex. Next, I focused on the next step is to the emergence of the vegetative cell. So here this is just before the emergence of the vegetative cell. So we could see some the cell wall, degrading cell, rodlet layer. Also, we could see some degrading cortex, and with the hint of a nascent vegetative cell. So we could see some structure, knob-like structure on the surface of the vegetative cell. So the last stage of the germination is the hatching of the new cell. And at this stage, the coat layer degrade, and still it's not detached from the cell, the new cell, and we could see the cell outside the coat layer. So this is the result, and we could see that this is the budding or hatching. So this is a rodlet layer, and this is the new cell. So brown static indicates the rodlet layer, and the purple static indicates the vegetative new nascent cell. And there are some knob-like structure on the surface of the vegetative layer. But as the cell grew longer, we could see that there is a structural change on the surface of the nascent vegetative cell. So these structures are filamentous structures appeared on the elongating nascent vegetative cell. So this is the last stage where the rodlet layer still attached but fully degraded, allowed to hatch the vegetative cell out of the it allowed to hatch the vegetative cell like this. So until now my conclusion was like I could see with the help of quick freeze many new structures. For example, I could see the native structure of crust layer, and probably its function is adhesion and resistance for the spore. And then I could see some outer vesicles, these green indicated with this green triangle, and these vesicles are just outside the core membrane, and they can be rodlet remnant, or maybe they have some other function, but still we need some future experiments. Next, I could see the spike-like structure, the native resolution, and these spike-like structure also provide resistance. And last but not the least, we could see the surface of the vegetative cell, new vegetative cell. So further, it will help for cell division and the growth of the cell. Next, beside these new structures, I could also see the improved structures. So one of them is cortex. The cortex is very important as it involved in the degradation of, it involved in the protection of this core. It is believed that the cortex provide the protection to the core and involved in the dormancy of the core. Next, I could see the core membrane at high resolution at both during the endospore and also during the germination process. During the when the endospore I observed by using quick freeze, we could see these vesicles are very kind of like normal vesicle-like structures, but during the early stage of germination, these vesicles integrate and disintegrate and kind of merge with the membrane. And next is, So I could also see the core membrane expansion, because in the core membrane, in the endospore, the core membrane is it has a specific volume and area. But when we notice that its area expands drastically during the germination process. So this is very important information. Before this, it was visualized by many techniques. So this time we just improvised the resolution of these processes and these structure. So I acknowledge, thank you so much. This is my horizon. So I acknowledge my lab mates. Special thanks to Professor Makoto Miyata. He has been a very supportive for my whole PhD career, and also a special thank to Yuya Tahara and Junko Shiomi and my very good friend Hana Kiyama, and my family, and of course my parents. Thank you so much.
Thank you for the presentation. So now the floor is open for questions. What priority will be given to the questions from the discussion committee members?
Yes.
Question, okay. During germination, cortex structure degrade. But you said lysine did not completely degrade peptide glycan of spore.
Yes.
Yeah, during germination, can you explain what type of enzyme degrade peptidoglycan of protects the spore?
Yes. Actually, there are some specific cortex specific, degrading specific enzymes. They are present inside the pore, but the lysozyme is outside, and there's not lysozyme. They are different from lysozyme, so they are very specific to the cortex. For example, one of the example is the CLDW amidase. Kind of it degrades the cortex. So when the germination occurs, the cortex from the inside, the enzyme from the inside of the pore goes outside and degrades cortex. But we are talking about lysozyme, which is we are providing from the outside, and it's not the enzyme inside the pore. So those enzymes which degrade the cortex are different. They are not lysozyme.
Okay.
Yes.
I have a couple of questions.
Yes, sir.
The first is a very fundamental question about the life cycle of batches such, and what triggers sporulation and what triggers germination. What is the important determinant?
Sorry, something?
And what is important determinant of germination of batches? And what is an important determinant of sporulation?
Okay. Thank you. So actually the germination, the most important determinants are the peptides, amino acid or anything, and their peptides are like when they connect with the GR receptors present inside. For example, I will so I'm just telling you myself. So actually when during the when the spore come across some kind of environment like nutrient deficiency, like kind of signals through phosphorylation, they get the signal by phosphorylation and they find out that there's no nutrients. So they actively change their mood to the sporulation. But during the germination, the inner membrane is the determinant of the germination because it contains the GR receptors, which immediately find the signals such as peptides and amino acids, and they drastically changes their process to germination.
And you showed a very interesting images. And I'm wondering the role of these structures, for example, crust and vegetables. Do you have any idea?
Load?
Role, yes.
Means
role of crust and the role of vegetables. Yes.
Actually, I tried different concentration of the lysozyme on the crust layer, but only the number of the cell decreases, I mean the spike decreases, but the structure remains same. So I think the structure doesn't have any, like the lysozyme have the same effect, no lower, yeah, the same structure, yes.
Hair-like structure or spike is partially degraded by lysozyme.
Yes.
Can you explain the difference in composition of lysozyme degraded
Lysozyme degraded.
but difference?
You mean to say that they are degraded by the lysozyme? Actually, the crust, when I treated the crust with the lysozyme, so it totally changed its structure to spike. But the degradation we see in the spike is probably because of some components of that crust or maybe artifact related to the, like for example, sometimes we do some quick freezing, and during that we could see some artifacts, kind of unnatural structures. But I think the major difference is lysozyme act on the peptidoglycan because it has some kind of components. For example, like glucose and amino acid or something. So, for example, there are a lot of glucose involved, same glucose involved in the crust. So when the lysozyme act on the crust, some of the components which are common with the spore peptidoglycan are not affected by the lysozyme. So we see some changes in the structure because of that.
I'm interested in the hair-like structure. Is that composed of the polysaccharide or protein polysaccharide complex?
It's kind of both protein plus polysaccharides. Yes, it has both proteins and polysaccharides. There are a lot of important proteins involved in the hair composition, and when we like previously when the scientists mutate those proteins, so it affects the polysaccharide also. So I think they are connected with each other because when they degrade, when they make the mutants of those proteins, so there was clearly they were affecting the polysaccharide also. So I think both.
Is the structure specific for spore?
Yes, only actually there are different species of bacillus which are spore forming, but the Bacillus subtilis structure is different as compared to the other one. The other outer layer is also hair-like structure, but with extra part, like kind of space. But in the Bacillus spore, this crust layer is tightly attached to the surface. So there's some difference between the proteins, but mostly the structure is same. And the one major difference between the Bacillus subtilis spore and the other spore is that by the other techniques, they can't observe this structure without staining. But the other spores, for example, Bacillus, Anthrax or Clostridium, they can be easily visualized by negative staining or something. But Bacillus subtilis spore, it was difficult, so they used ruthenium staining. But in our technique, we could see without staining.
いかがでしょうか。目が悪いので押してあげてても吐き上げてもないとわからない。
はいはい。セルボールストラクチャー、ネットワーク、こういうなんちゅうサーフィス、バット、ヘアライクストラクチャー、なんちゅうの、フィラメンタス。
Yes.
だからフェアライクストラクチャー、ベリーインタレスティング、バット、バイオシンセシスオブフェアライクストラクチャー、can you predict or think, explain?
You mean to say that the structure is different from the other spore? Actually, the other spore, for example, the next second layer, the rodlet layer, composed of 70 proteins, and each layer is attached with each other somehow, but the outer crust layer is very different. If we remove the outer crust layer, there is no effect on the rodlet layer. And similarly, if we remove the rodlet layer, there is no much effect on the core. The core still germinates. So each layer has their own specific proteins. Somehow they are related, but not functionally. They are not related. They will function the same without each other. So that's how. Their crust is very different as compared to the rodlet layer.
Okay, I have one question.
Yes.
You said the hair-like structure may be developed in the adhesion and
Yes.
resistance.
Yes, yes.
I understand the polysaccharide layer
Mm.
is involved in the adhesion.
Yes.
But how the head structure contribute to the resistance?
Yes, actually, I think I'm not fully sure, but previously some researchers have experimented and they digest the spore with protozoa. So protozoa digest the spore and they isolate the spore from the protozoa, and still it was not affected. So they speculate that this is because of the outermost surface, but I think because it consists of some remnants of PG, peptidoglycan, which is highly resistant, so that's why I think it also protects cell from spore from the other thing, other than the adherent, because it's a mixture of proteins, spore G proteins, glucose, and a lot of for example, polysaccharide. So everything performed differently according to the requirement. That's, I think, is my assumption. Thank you.
Okay, it's time to end. Thank you. どうもありがとうございました。
審査員の先生、ちょっと残っていただけますか。